And so Dr. Liu is currently a professor in the Department of Medicine and Biomedical Engineering at the University of Pittsburgh. And she received her master from University in chemistry from University of Chicago and her PhD in biomedical engineering from Northwestern in 2006. And then I met her as well when she was doing a postdoc at Johnson & Johnson. And then after that, she instead decided that industry was not her cup of tea, and she moved back to academia. I don't know. I don't know about that choice. In 2008, and she started her own lab, and she's an extremely successful researcher with many accolades, including she's authored more than 80 peer-reviewed articles, many of them in Nature and other very prestigious journals. She also has many patents under her belt, 10 patents. A lot of funding from the National Institute of Health, which is real mark of success. She's doing it, and she's internationally recognized, had won many awards, and including being the fellow of SPIE and of Optica, and she won the PIC Innovator Award twice, and the Hillman, and as well she's a Hillman Fellow for Cancer Research. So I introduce you, Dr. Yang Liu.
Thank you so much for a very kind introduction. It's truly my great pleasure to be here. So my lab is working on I'm a microscopist, so we develop optical microscopy techniques to address unmet clinical needs in precision medicine. Or my main area is actually in cancer research, so primarily focus is in cancer. So I think most of you are probably familiar with precision medicine in the context of cancer. People call it called precision oncology. So the first thing probably comes to your mind is about finding the right treatment for the right patient. There's a clear need there because typically a therapy, only 20-30% of patients will respond with a specific type of therapy. So there's a clear need for precision oncology. But there's also an area of precision medicine that's less recognized. It's called precision prevention. So the idea is because most human cancers takes about 20 to 30 years to develop. So that's really a long window of opportunity for us to prevent them before they become malignant. So unlike the cancer patients, so the main population that we deal with for precision prevention are those at-risk patients. So these patients have increased risk for developing cancer, but they don't have cancer yet. So these are patients like Barrett's esophagus, ulcerative colitis, or those with colon polyps that they find some precursor lesions, or high-risk breast lesions. So those are just some of the examples. So here, the biggest challenge is most of those precursors are perfectly fine. They stay benign throughout the entire course. So, but only a few percent of those that really at high risk and likely to progress into malignancy. It could be 5-10 years down the line. The clinical practice is basically to do surveillance on all of those patients, millions and millions of people with a precursor or with these at-risk conditions. So there's actually a clear need for precision surveillance to prevent cancer. So the goal is basically identify those small percentage of those patients that's most likely to develop cancer. So we can focus our healthcare resource on those that truly need. So that's basically the clinical problem we've been trying to address for the last 10-15 years. So the key is really to understand why some precursors that eventually develop into malignancy. So there's a need to understand the underlying cancer biology behind it, and there's also a need to develop technology to address those issues. As an engineer, one of the first things to address a clinical challenge, you talk to a clinician. You want to understand the clinical workflow. So we work a lot lot on colon cancer. Colon is actually probably one of the best targets for prevention. Because there is a colonoscopy done on the general population, and colon polyps is very common. I think it's up to 50%. The statistics I read, about 50% of those procedures will end up with a colon polyp. And then some of them are precursors. So what the physician do is, regardless, whether it's malignant or pre-malignant, they'll remove them anyways, and they embed them into this paraffin block, and they store them for decades. So if you think about, what is the best strategy to sort of develop technology that fits into the workflow, is actually use this paraffin embedded tissue, because they are collected as a standard of care. You don't really need a clinician to go back and collect extra samples. So that's really the best scenario. If we can take advantage of these and extract valuable biomarker from those tissue, that's really the best integration with the existing clinical standard. So what the clinician currently do, the purpose they're taking the tissue is they will take a section of it and they stage H&E, and they look for basically the abnormality in the tissue architecture. And at the single cell level, they also look for the morphological changes of the nucleus. So there are some sort of a list of characteristics they're looking for, like the size of the nucleus, or the shape of the nucleus, and then the texture of the chromatin. So those are the nuclear architecture within the nucleus. So there are certain criteria that can be used to define precursors that are called adenoma or dysplasia, or there are certain criteria that can distinguish, identify, classify it as adenocarcinoma. So that's a gold standard that's been used for actually almost 200 years. The one thing that always strikes me is those nuclear morphology, the characteristic changes, the physical changes in the nucleus. That's really ubiquitous. So in cancer, we talk about driver mutations, a lot of the molecular heterogeneity. But these physical properties of nucleus, that's really ubiquitous, those characteristic changes, is almost universal across different tumor types. So that's kind of intriguing. In diagnostics, sometimes it's actually valuable, especially at the early stage, to find those common features independent of molecular heterogeneity. So obviously, this microscopic changes is not sensitive enough. So let's get to the molecular scale. If we want something more sensitive, we have to get down to the molecular scale. With all this research, decades of research in cancer biology, molecular biology, so we now know, within the nucleus, we know there's DNA there, but DNA, so human genome is about 2 billion base pairs, and DNA, if you stretch straight, is about 2 meters long, but they have to really fold it inside the nucleus to fit into that physical space of a few microns. So that's really 6 orders of compaction that those DNA has to undergo in order to maintain that function. But they are not random. It's actually highly compartmentalized. So when they fold it inside the nucleus, some regions they're highly condensed, some regions are open. The reason they have to assume those different level of compaction is because of they regulate transcription, and they can turn on certain genes, and they can turn off certain genes that regulate gene expression. So that's clearly a very important event for cancer development. Another important event is that only 5% of the genome are protein coding, 95% are non-coding regions. So in the past, people call it junk DNA, but it's actually not really junk. The compaction of those also maintain the stability of the genome. So that's another biological significance to look at the compaction of the chromatin. So that's basically, what we are trying to do is design technologies that can detect those molecular scale changes of chromatin compaction that's important in carcinogenesis. And but if you look at it from an engineering perspective, right, it's not just one area of looking at molecular changes. It's across the scale problem. So every single event, all the way from the tumor, the millimeter, centimeter, or millimeter sized tumor, to the architecture of the tissue, to the morphological, to microscopic changes to molecular scale. So all of these are important in that specific context in the clinical scenario. So light microscope is really the central tool for this task in the clinical care. So the conventional microscopy typically work within a wide range from centimeters all the way down to the diffraction limited resolution. So that's roughly 250 nanometers. So that limitation is no longer the case in the past decade because of the development of nanoscopy. So that's basically using light to detect molecular scale structures below the diffraction limit. So the capability of light microscope really expanded to 7-8 orders of magnitude. So in my view, the light microscope has never been more powerful. So what's the future lies is our ability to connect these different scales. And so we can have a full understanding of the pathological conditions, whether it's precursor or tumor in the case of cancer, to understand it from molecular scale all the way up to the microscale. So we developed technologies that kind of circled around this goal of multi-scale imaging. So my lab has done quite a bit of technology development from the mouse doesn't work. Do we have a pointer?
Pointer won't work.
Oh, won't work either, and that's okay. I just moved.
I think there's another one here.
Because I connected with my computer. It was working before. Anyway, I just keep going. So we've been developing techniques that's kind of multi-model. So in early days, we worked a lot on quantitative phase imaging and detecting sort of the nanoscale architectural changes using interferometric technique. And later, we have developed the super resolution microscopy as a support to understand the changes in the nanoscale nuclear architecture. And then more recently, we kind of developed a cross-scale system to have a full understanding of the tumors or precursors in a wide range of scale. So one of our main applications is in cancer prevention and detection. And besides that, we also work on a wide variety of both basic and clinical translation problems, such as looking at drug resistance, assessing mucosal healing or wound healing in the colon, and then looking at the chromatin biology. So today, my talk I'm going to introduce the technique we developed and give you some examples for some of the applications we apply those technologies. So let's start with phase. So why we use phase, right? So, and the idea here is, for those of you not familiar with this, so if you have a plane wave that illuminates the samples, and because the light travels at different speed, because of the refractive index, they slow those the waves down. And that introduced the delay on the wavefront. So the regions that interact with the cells that get delayed. So those wavefronts can be detected using either interferometry technique or computational imaging to recover it. So that, even though the image, for imaging technique, the image is still diffraction limited, but that value of phase actually carries the nanometer scale information about the subtle changes in the architecture. Basically the information they provide is in the transmitted direction is the mean refractive index or the shape of the cells. We call it morphology. And we want to integrate it with the clinical workflow. So we developed this paraffin embedded. So basically standard paraffin block, and we take a section of it. And we want to minimize the complexity of sample preparation. So that's a standard pathological processing. And then in the case of phase imaging, so the light transmitted through the sample in the transmitted direction, and then we detect it through, this is the detection part of the optical microscope. It's a fairly simple setup. The diffraction gratings that separate the wave into the zero order as well as the first diffraction order. And then the zero order essentially is the background. And then the first diffraction order is the scatter light that coming from the sample. When they interfere on the camera, they interfere at a certain axis with a specific angle. So this is so-called off-axis digital holographic microscopy, or DPM. And this specific configuration is called diffraction phase microscopy. So what we do here is, so if you look at the interferogram on the left, that's basically the fringe. You will see those fringe that coming from the interference effect and the spatial interference. So if you look at that term up there is delta K times X. So that basically carries that spatial interference. The reason we want to have this off-axis interference is to have, so we can separate, if you take a Fourier transform, you can separate the zero order and the first, the different diffraction order. So we take the interference term, that's basically that carry this cosine, and we then take a Hilbert transform to recover the phase. So if you look at the image on the right, that's basically the quantitative phase information that we can recover from this kind of setup. It's very simple snapshot acquisition, but you can get quantitative information. So the application we use it for is basically trying to improve the diagnostic accuracy of cytology. So these are basically, urine cytology is commonly used in sort of patients with bladder cancer or suspect they may have bladder cancer. So that's actually one of the hardest diagnoses that pathologists have to make, because there are lots of sort of ambiguous diagnosis sort of in the middle group. But with the quantitative phase, because it's highly sensitive to the morphological change of the cells, so we thought, by providing that objective measurement, that could potentially improve the diagnostic accuracy. So this is a study we did actually several years ago, comparing sort of 4 different categories that defined by the pathologist. So negative, atypical, suspicious, and positive. So you can clearly see the information from the face, the image on the top that show the separation. So you see the difference between the nucleus in the center and versus the cytoplasm. And then they show distinct image contrast in those different categories. And if we look at this quantitatively, so the figure on the upper left are those extracted from the entire cell. So one parameter you can extract from the quantitative phase, besides the phase value, we can also integrate times S here is basically the area. So the optical path length are measured from the quantitative phase times the area that give you basically the phase volume. And then the alpha there is basically just a constant that will link the phase volume with the dry mass. That's really related to the mass of the cells in the unit of picogram. So if you look at the figure on the upper left, that's basically the dry mass from the entire cell. And then the one on the right is the dry mass of the entire of the nucleus. So you can see that the cell dry mass can distinguish the suspicious or the positive group with the negative group. So the atypical is closer to the negative. But the nucleus dry mass can really distinguish, correlate very well with the clinical definition of those 4 different categories. And if we plot this, if you look at the figure D, the one on the bottom over here, you can see by plotting the entropy, which is based on the phase map that you can calculate entropy versus the cell dry mass, you can see the distinction of those 4 different groups. So we can develop more quantitative assessment for this psychological diagnosis. And so that's a phase in the reflection, in the transmission configuration. But there's also, when the light transmit, when the light properly interact with the cells, a portion of the light can get reflected. So that is basically capturing the high spatial frequency component, the finer structures of the cells. So if you look at this, the cell gets reflected, and this is kind of gets similar to the optical coherence microscopy. So they interfere with a reference beam. That's basically, again, we didn't change the entire setup on the same slide, and we collect them. So when we collect them as a function of wavelength, so this is similar to the spectral domain OCM, so it's a full field optical coherence microscopy. And we can use a tunable source to change the wavelength and collect the spectral interference signal. So what that means, right? So that's basically the equation that describe essentially all the interferometric techniques. Just kind of ignore all the main details. So we can extract from the equation here, if we take a Fourier transform of the spectral interference signal, we can extract the phase. But because in OCT or in this kind of interferometric setup, and in the reflection direction, there's another level of complexity is the coherent gate. So essentially, we know, I think probably most of you know about OCT. And in OCT, basically you're detecting the strong reflections that are coming from the samples. So if you have a strong mismatch, or they call it interface, in between different layers, and you can, after the Fourier transform, you get a peak. And if you take the phase value at that optical path length locations, you get the phase value here. So if you map this pixel by pixel, you actually can get this phase carries nanoscale information. So you can map things like nanostructures. But in case of the sample we have here, it's actually not, we don't have the strong interface. We actually direct all the strong reflections somewhere else. So what we have is a special scenario. It's just weakly varying refractive index that coming from the structural changes within the cells or the tissue. So what you get here is still the optical path length profile. It just didn't show those strong reflection peaks. And then because of this is a low coherent light source, so they convolve with basically the coherent gate. So basically what you end up getting is this coherent gate because they have a depth resolved capability. So you can kind of do a sliding window digitally and probe the structural changes inside the nucleus. So it's kind of theoretically, so this is, the OCT theory has been well established, but typically people deal with sample tissues with strong refractive index mismatch. But the slowly varying refractive index mismatch is actually a tricky problem mathematically. So my former postdoc, Shikhar Uttam, so he's now running his own lab. He developed the theory to interpret this signal. So you have the optical path length, and that's basically the phase term. And the one component we call it sub-resolution offset, so it's the delta COP out there. So that term essentially give you, it's kind of similar to the phase-sensitive OCT that detect the changes of the sub-resolution, the optical path length change. But there's another term here, we call it the mean spatial frequency. So if I expand it, so for example, if you have a refractive index profile transition within the samples, and this is reflection, that's actually the big distinction between the reflection QPI versus the transmitted QPI. Is the reflection QPI doesn't detect the average refractive index. So this is basically what's captured by the transmission information. You don't see that much change that captured in the reflection configuration. So if you look at the amplitude, the range of this refractive index change, it's also very, it's not a dramatic, there's some variation as you increase the delta N, but it's not a strong dependence. So the biggest change actually coming from this sort of the spatial frequency or rate of change. So whether you have a sharp change or slow change at a specific location. So that's actually showing the strongest dependence. So just kind of summarize, if we take those spectral interference signal, and then we take a Fourier transform and extract the phase at certain oversampled depth, and then what we end up getting this is a sort of, if you have a refractive index profile like this, if you have a slowly changing versus a fast changing refractive index profile, those fast changing profile will give you a higher delta OPD value. So that's kind of the physical measurement. So essentially what they detect is the depth resolved local changes in the refractive index profile. So we integrate this both in one system. So we have a broadband illumination. So this configuration here detects the reflection phase, and then this detects the transmitted phase. And this is basically the standard bright field imaging. So everything incorporated into one system. So this kind of gives you an idea of the different type of contrast. So the top two the figures, so the one on the left is bright field, and then on the right is the quantitative phase in the transmission direction. So you see that high contrast from the cells. And these are the cells basically embedded in the matrix. So you actually can, based on this image, you can tell the matrix is very thin. So that's why you barely see it in face value is much lower in those images. But the reflection QPI, the one that give you the depth resolved information. So if you look at those three images at the bottom, you can see the one that close to the substrate, and then you move up. And the one that close to the substrate is where those matrix located. So you can see the images on both images on the first step, but not on the second and the third. And just kind of give you the complementary information here. So here, this nucleus, these cells, you can see on all three depths. But the one on the top, you only see on the top two. So that means the cell is thinner. And if you look at the quantitative phase map up from the transmission QPI, so you can see the one on the top, that's thinner. So you see overall less cumulative optical path length compared to the one on the bottom. So that's kind of really give you a different type of contrast with those two different configurations. So these are the comparison of tissue, H&E standard, the stain, and then the QPI in the transmission direction. And that give you a really nice view of the tissue architecture. You can see some cellular level information that coming from the higher refractive index, like the immune cells in the extracellular matrix, or some inflammatory cells. And then the one on the bottom, it looks more grainy because of this detecting local changes and doesn't have those cumulative contrast that you would normally get from the transmission QPI. So what we want to do here is we want to look at the architectural changes, the local changes in nuclear architecture that just coming from the nucleus. But you can see the phase image, it's actually hard to get that contrast. So you need H&E to get the nucleus information. So the way we did it, we developed a workflow to extract that nanoscale nuclear architecture. We call it NanoNam. Essentially, it's the integration of all approach. So we start with unstained tissue, and then get a high contrast transmission phase image, and then this low contrast reflection phase. And we do H&E staining, and also get the high contrast phase image on the stain. And those two image, stain and stain, we can co-register them, get a coordinate, and segment the nuclei from the standard H&E image, and then apply that back onto the reflection phase map. And that gives us essentially the nuclear architecture map just from the nucleus. So that's kind of the final outcome you see. And the information we use for quantification, essentially those bottom ones that give you the local changes of nuclear architecture. So that's one of the clinical studies we did, is to look at patients with ulcerative colitis. So that's an inflammatory condition. And that's actually the value of optical technique to assess archive samples. So these are the samples stored, and then we have the clinical outcome. I mentioned earlier, a lot of the precursors that really take 5-10 years to really see the outcome. So the ability to go back to the old sample actually gives us lots of value in this kind of studies. So this is a 2-patient One never developed cancer, one developed after 7 years. So if you look at their nuclear architecture, the map, you can see the ones, the high risk that give you a much higher value. That indicate, I'll show you exactly what the actual structural changes later. But that's what this physical parameter tell you is basically the local changes of refractive index is more heterogeneous. So if we look at our expanded,
I'll ask you a question.
Oh.
How do you know that the storage of the sample doesn't affect any of these characteristics?
So it's a great question. So these are, so we actually looked at the dependent on sample age. So if you look at traditional immunofluorescence staining or IHC, the histochemistry, the epitope could get damaged. These are DNAs, it's actually very stable. So within the 10 years period, we compare those at different age, and there's probably because of the DNA we measure, it's actually stored.
That would take a 10-year study though, wouldn't it?
No,
How did
it's
you
just
do
the
that?
archive. You just go back and you look at
And you can't look at the same sample to
Yeah.
see if it's changed over 10
not
years.
the same sample, the same category. Like say if you look at controls and that's probably the best you can do. Otherwise you have to do that for 10 years, yeah. But for proteins and RNAs, especially RNAs and the protein epitope, it does depend on the age. So this is, so if you look at the high risk versus low risk, you can see that overall statistically, the reflection phase value is higher. And then if we spread it out, the mean value versus entropy, we can see the separation between two different patient populations. And here is just a Kaplan-Meier curve that shows the time to progression. So if we take entropy and then we define a cutoff line between the negative and positive, so we can separate those two as well with high level of statistical significance. And then moving forward, this is a prospective study. The patient get followed for 5 years. Because that's typically how long that grant normally runs, about 5 years. So you need a renewal to do a larger scale study. So this is, we look at the both low risk versus high risk. And so here, because we get the DAX resolved information. The previous analysis, we only took the average. Here, essentially, we're just, we thought, we should take advantage of the entire 3D information. Because, you have that coherent window we can slide. So what we did here is we take a different optical path length and then map it out into a matrix here. So each of the column essentially is a nucleus. And then you have this gigantic matrix, and what we did here is basically extract how we can account for all the information. So Shikar did, what he did here is he had mass training, so he uses common analysis that using the matrix analysis. He calculates 3 statistical parameters, the spectral norm, 1 norm, and infinity norm. So this is essentially the calculation that's coming from the entire matrix that accounts for both internuclear and intranuclear variations within, across all the patients. So if you look at this curve, the red curve that comes from the high-risk patient, the blue comes from the low-risk. So you can see the shift of that high-risk patient. So that's consistent from the earlier studies we did on the retrospective samples. So if we project all these 3 parameters in a unit sphere, you can also see that separation between the low-risk and high-risk. So we did this with the training set. We split the patient into half, and the training set with the area under curve about 9.93, and then we also did a validation set on the independent patient cohort. So with area under curve of 0.86. So that shows the promise of this approach. So basically, with the quantitative phase imaging that's captured in both transmitted and reflection direction, and both are interferometric techniques, and we can measure the refractive index changes with nanoscale sensitivity. So we integrate this approach and develop this nanoscale nuclear architecture mapping that shows the potential to basically identify high-risk patients in early stage precursors. But they kind of get to the third point. The first two, is more of a physics engineering base. So when you explain this to a clinician, no one really understands exactly what, you can develop all the theories, no one still, the biologist, either the biologist or clinician have no idea what the refractive index really means. So to really get this thing across, I said, people ask me, you talk about all the structural changes, what the changes are. So that kind of get to my second phase of the development to understand what are these molecular scale changes in the early stage carcinogenesis. So the timing was, actually about maybe 8 years ago, we started this technique that kind of leveraged the success of super resolution microscopy. We thought, it's the right timing to really look at those nanoscale structures that would be difficult to probe in the past. So there, with your coursework, you probably heard a lot about super resolution imaging technique with different acronyms, SIM, STED, or STORM, and POM. So we focused on the single molecule localization base, or the technique like STORM, POM, sort of fall into this category. So the idea here is it's actually a fairly simple concept. So we know the resolution of the optical system is diffraction limited. So that's why if you look at the point spread function, and it has the width of those is about, so it's kind of like a Gaussian shape. The width is roughly 250 nanometer, I mentioned earlier. But the peak can be identified at a very high precision. So it's a kind of like a mathematical feeding trying to find the peak location. So if you have a sort of a fluorescently labeled samples like this, you don't know where the individual molecule is. So that's where some of the chemistry technique that come into play. So the idea is if I switch off most of the fluorophores, I only allow a small subset of those to turn on. And then you basically just repeat that process, accumulate those dots, and eventually you can reconstruct the image. Because you're kind of focusing on the center of those point spread function, you can get a much higher resolution compared to the conventional technique, about the improvement in the factor of 10. So just kind of give you, again, so switch it off and then you localize the center and you repeat this process. It's like a sampling. And then you can get a super resolved image. So the instrumentation-wise, lots of people think super resolution has to be something really complicated. It's actually just a wide field microscope. It's one of the easy things you could do, coupled with a high power laser and also with sort of a stabilization system, because you have to maintain a nanometer scale stability throughout the entire data acquisition process. So that's the only caveat there. And when I first started this, I wasn't really in the super resolution imaging field earlier, and then I thought this, the commercial system, I can kind of just use it, will be a fairly straightforward study. And that's kind of some of the early images you get. The images are beautiful, but the problem is it wasn't really designed to look at a sort of a clinically relevant samples like the tissue section. So I quickly realized that's, it's a nice instrument to generate pretty pictures, but when you go further to answer challenging biological problems, it's quite limited. The biggest problem is, one is low throughput. Early days, the imaging field of view is 25 by 25 micron. So you just see like sometimes 1 or 2 cells. So in our study, we typically need to assess hundreds of those nuclei. It's just gonna take forever. And the bigger problem is actually the image quality varies quite a bit depending on the quality of the raw data, because of the nature of this approach is computational image reconstruction. So the lack of reproducibility was a major problem. So we actually put in lots of effort, almost 5 years of the work, that we eventually developed the method. We call it PASStorm. It's basically a storm, which is a sort of a super resolution imaging technique. and then we modified, adapted for pathological samples. So again, we work with those paraffin embedded tissue, we take a section of it. So the problem of these is the raw data initially. So you get, a really high background, because the nature of this technique typically require high power laser. And you end up with really high background on the paraffin embedded tissue. So to address that problem, so what we did is a very simple clearing, just 5-10 minutes clearing with the refracting index matching. You can suppress the background and recover the single molecules that needed for super resolution. But it's still not, especially for paraffin embedded tissue, the background is really an issue. So we further use the post-processing, take the temporal step, and develop this background removal method. Basically, completely remove the background from the raw data, always left in those individual single molecule to recover the raw data. And because with the upside of removing the background is you also remove a lot of noise. So that's actually led to the next method that we developed using a deconvolution method, a linear deconvolution that's insensitive to background, to the noise. And previously to reconstruct, so this is basically a filtering. So we take a Fourier transform, we truncate all the noise, and recover, do an inverse Fourier transform. All these operations one-step linear without the iterative deconvolution method. So the computation speed is much faster. So the previous data that oftentimes took hours or sometimes days to reconstruct, now it's done basically online. So the definition of online is the image reconstruction speed is faster than the data acquisition speed. And then, so that's sort of eventually we get this much improved resolution as well as image significantly reduced image artifact. So that improved the reproducibility. So if you zoom in, you can see the improvement. The ones with the background corrections, with all the clearing, that's just much dramatic improvement in terms of just more clear background, much reduced image artifact. But it's still, the image throughput issue is still a challenge. So the reason that, lots of time in high resolution imaging, we go with a small field of view. The reason is, if you look at the laser profile, the illumination, that's a Gaussian shape. But typically we prefer a uniform. That's especially the case for the storm-based approach, because otherwise you get different density with different blinking rate. So, and people typically take the center to truncate the edge, so the intensity at the center typically higher compared to the one on the periphery. So to solve this problem, actually it's simpler than most people thought. So we basically just couple the laser into a multimode fiber and then put the vibration. Like some people do the toothbrush, those vibrating toothbrush. So here we put a piezo vibrator, so we can turn this Gaussian profile into a flat field illumination. So that's, lots of time a solution doesn't have to be complicated. And these turn out that expand the field of view by roughly a factor of 6. So we can use it to look at, to get about 150 nanometer, 150 micron field of view. So we can look at things like DNA origami, and this is the 3D super resolution images, and also the tissue section. So it's kind of give you a context. That's what we used to have. That's actually already expanded field of view of roughly 50 micron. If you go with, say, the Nikon second generation N-Storm, that's about this field of view, about 50 micron. And we expanded further to about 150. And one important aspect is not just make it faster. So I used to send those, the small field of view image to pathologists and ask them, are these precursors, what type of lesion it is? They always come back to me and say, I need a whole slide. I can never tell with that tiny field of view, because they need the spatial context, that architecture, to identify the lesion. So that's not just for the speed, it's also for the accuracy of the type of diagnosis. And so that that moment, I start to recognize the importance of tissue architecture that's required for the clinical diagnosis. So that's kind of our first effort to get the cross scale. So this is 150 micron, and the resolution, if I expand it, the resolution here is roughly 30 nanometers. So this is about 5 orders of magnitude across. So that's needed because we need both the information of tissue architecture and the molecular scale information. And later I'll show you we expanded even further with 2 additional orders of magnitude. So with this approach, we start looking at, this very first question I had for years, what are the architectural changes that's really happening at the early stage, those things that are happening at a nanoscale. So we use this animal model here. So this is the APC minimized. And for those study colon cancer, that's one of the most widely used animal models. So we look at 6 weeks is just normal morphology of the tissue. So it's a very early time point. And these mice eventually will all develop tumors at the end. So at 12 weeks is the stage that you can see some tumor cells. So, and every time, because our goal is to correlate this with the refractive index measurement we looked at earlier. That doesn't have the specificity. You don't know whether you're looking at DNAs or RNAs or proteins. So we have to look for something predominant. So here we basically focus on the DNAs or the condensed forms of chromatin. And that's because that's a more predominant, that can give you stronger changes in the refractive index. But labeling DNA, I'll show you in a few slides, but labeling DNA is actually quite tricky in the super resolution condition. So we actually label the DNA binding protein here using immunofluorescence staining, and we target this condensed form of chromatin. And this is kind of, because they're enriched in the condensed form of chromatin, so each of these is a nucleus, if you look at the second row here. And those regions with sort of a foci, and that's the so-called heterochromatin foci. Those are really the condensed regions of the DNA. And this is visible, so the left, the red figure with color-coded red, and that's a microscopic image, diffraction limited. And so you see those heterochromatin foci as well. But with the super resolution, we actually can zoom in. Those big foci, you don't see the detailed structures because of the limited resolution. But super resolution, you actually can zoom in, look at how the sort of the building block of those microscopic foci. You see 6 weeks, that's normal morphology, but the nuclear, the heterochromatin compaction already more disrupted. So you see some of the dissociation of those spots. That's basically the heterochromatin domains that formed at the nanoscale. So if we quantify this is, we did this clustering analysis. That's a Gaussian clustering. We automatically identify those individual building blocks and then calculate the size. So, and we get the average per nuclei, and this is the figure that each of this group, roughly 200 nuclei per group. So you see a very dramatic, it's actually even in 6 weeks, that's normal cell, just with driver mutation in those cells. You can clearly see the chromatin already disrupted, and with the smaller cluster size. And this is continuous, so they keep changing throughout the entire process. But sort of the rate limiting step, kind of at the beginning, once that driver mutation is there, that cells or those mice that destined, because eventually they're all going to develop cancer at the end. So that rate limiting step, you see a biggest change in the heterochromatin cluster size. So again, this is just a different way to analyze this is a correlation map. So you see consistent result with the less correlation distance as the carcinogenesis time progressed. So that's kind of get to the second validation. The previous one, based on immunofluorescence. So if you do a lot of antibody labeling, you're always concerned of the penetration of the antibodies. So the biologists always question whether the thing you see is because of the artifact of the labeling. So this is a different validation. As I mentioned earlier, there's not really good dye for storm imaging that targeted sort of the small molecule DNA binding dye. So in collaboration with my chemistry colleagues at Vanderbilt, and they basically helped me develop this conjugated dye that's actually previously shown used in other contexts. The host, which is a DNA small molecule DNA binding dye, coupled with the Cy5, which is good for superresolution for storm. So when we conjugate this together, the host would get delivered into the nucleus, and then the Cy5 is used for imaging purpose. So when we have these, we can directly target DNA. And you can see again, the normal cells, they are very densely packed, predominantly in the periphery of the nucleus. And then when we move to the precancers and cancer, so the precursor, you see some disruption at the periphery. And then when they move to the cancerous tissue, you see almost a redistribution. You barely see that very dense patterns at the periphery of the nucleus. So that's kind of, if you look at the corresponding diffraction limited image, they have some indications, but those kind of image, typically when you see it, you cannot really conclude because of limited resolution. So the quantification again that show you the changes in that nanodomain size that's consistent with the previous data that labeling the hypochromatin. So these are the lesions that's identified by the pathologist. So the next question, especially in the clinically relevant context, is for those lesions that's pathologically the same, can we see the differences in the risk? So that's basically, it's still an ongoing project, but the initial proof of concept, so this is in collaboration with my colleague, Rocky Schoen. He specialized in colon cancer screening. So he collected a lot of those colon polyps with the documented clinical outcome. So what we compare here is adenoma versus advanced adenoma. So if you are in the field, so this definition is solely because of the tumor size of advanced adenoma is bigger. I think it's larger than 1 centimeter, but pathologically they're the same. The pathologist won't be able to tell the difference. The reason they call it advanced adenoma is larger tumors or larger polyps, they tend to have a significantly increased risk for developing colon cancer. So it's kind of served as the first step proof of concept. So we look at both adenoma versus advanced adenoma. So you can see, again, those at a significant increased risk, their chromatin already are more disrupted. So you see a smaller cluster size. And this is another project that we are working on with Dr. Pravinder Singh. So he's a chemo prevention person. So he basically evaluates the efficiency of preventive drugs. So, and what he did is he treated the mice with sulforaphane. So these are the drugs that's actually very enriched in like broccoli or some of those vegetables that's shown to be very effective preventing cancer. So when we compare this, so you can see those that treated with sulforaphane, the chromatin seems to be more condensed. And if you look at the statistical analysis, you can see even the same lesions, those that treated with the sulforaphane, the chromatin structure become more condensed. So what this tells you is, so it's actually two things. First, we talk about this compact chromatin structure is needed and sort of present in the normal cell. And here with the treatment of chemopreventive drug, they can be partially restored. So that means it's a reversible process. And the second thing is, essentially this shows this correlate with risk, right? So when you have a reduction of risk with the chemopreventive drug, you do see the chromatin structure that correlate with this. So that's another layer of validation compared to the data we had before. And this is another project that's also ongoing. We look at early detection of lung cancer. So the idea here is, lung is actually very difficult to access. But one of the samples that you can get that coming from lung is sputum. So that's especially the case for heavy smokers. That's basically the high-risk patient that should undergo CT scan for lung cancer screening. And sputum, this actually the sample. So my collaborator here, they collected those sputum samples over the years. And this actually very simple home collection. They gave patient a jar, and then the patient actually coughed toward that jar over 3 days period to get enough samples, and then they process it. So this is actually a very valuable resource for us to develop strategies for lung cancer detection. So the sputum just give you an idea, the upper respiratory system, there are cells that can be shedded into the sputum, and we focus on those cells. It's called bronchial epithelial cells. So this is basically initial study. We actually look at first get the tissue from, it's not the sputum sample, but the tissue, and we focus on the bronchial epithelial cells. So if you look at the healthy control versus patient with the cancer cell from the lung cancer, you can see that they're clearly separated. But in the sputum, lots of times you don't get cancer cells. The cancer cells may be far away. You don't really detect them from the sputum. So what this data is showing is if you have normal cells from the lung cancer patient, you can also see various degree of disrupted chromatin. So that actually gives us the promise that we don't need them to be all cancer cells. Normal cells can also capture that risk I was talking about as a potential for early detection. So we're actually just finishing up a 70 patient study. This is an initial analysis. You can see the patient with lung cancer indeed showing more open, more disrupted chromatin structure compared to the controls. So just summarize this part of the talk. We look at sort of using super resolution imaging, and we characterize the chromatin, nanoscale chromatin structure. And what our data is showing here is the disrupted hydrochromatin can serve as sort of it's happening at a very early stage of carcinogenesis, and it's a continuous process, and that potentially can be used as a sensitive marker for early stage carcinogenesis. Just kind of coming back to the refractive index changes I was talking about. So you see here, this is basically the goal is to correlate what we see with the super resolution imaging versus the quantitative phase imaging information we get. So you can see if we look at the same type of samples with both super resolution imaging and reflection QPI measurement, and we can see that they are indeed shifting, the higher risk, they're all shifting to a higher value. It's consistent with the previous analysis. So just in this context, you can see this is more abstract concept. So if you normal cells or the low risk have this condensed chromatin, when they become disrupted, if you relate to the refractive index profile, it's kind of like the course change of the refractive index become more heterogeneous because of those dissociation of this highly compact domain. And then if you think about what I showed earlier, you can clearly see the sort of consistent with the original what simulation is predicting. Just kind of summarize this part, the key finding here is this disrupted hydrochromatin could be used as a common feature. I showed the different tumor types. You can see it can be used as a common feature. But still have time? No? Can I have 5 more minutes? So, so that kind of, I talk about the need to expand the field of view, but we realize the limitation, so we need to further expand more to get sort of a systems level information. So that's get to the last part of my talk, and our recent effort developing this cross-scale imaging system. So when you look at the imaging system, a lot of times, people always talk about the magnification rather than the NA of the system. But in my view, magnification has nothing to do with resolution, right? So that's a sampling concept. So the idea is it's actually magnification is a bad thing. You lose your field of view. So the idea here is if I can use the highest NA possible, but it still have lowest magnification possible. If I couple that with the right pixel size, that's actually a more ideal system. So with this idea, we developed this sort of a cross-scale system. So this is a single field of view of 2 by 3 millimeters and at a sub-micron resolution. It's a fluorescent image. But why this kind of technique, why this kind of system is uncommon, is because when the field of view is getting larger, you don't get a flat view, right? You typically get its length is curved, so you get defocus at the edge. And not just that, you also have those aberrations. So if you look at the center, it's true. The image quality is much better compared to those ones at the periphery. So to overcome this, what we did here is actually a simple approach. We just take a few stacks, a few z-axis of the acquisition, and then we do everything post-processing. So this is the raw data, and the ones at the center versus those at the periphery. And if we refocus it, this is piecewise refocusing done all on the post-processing. So you can see a significantly improved, the focusing effect is better. And then with the deconvolution, we can correct for some of the aberrations that we see. So, and to couple this with the quantitative phase, so that's a fluorescence. We also want to incorporate the phase information. So here, if we look at so this is done through computational imaging. Because in this format, if you do an interferometric technique on this large lens system, maintaining the uniformity will be an issue. But it's actually quite straightforward. If you do a computational imaging, see the LED collimated beam that's coming from the top. That's the only extra component you need to add. And then we just take 2 defocus images and use that transport of intensity equation to recover the phase. It's actually a very simple approach. So we can get the entire phase image over this field of view. Again, we use that piecewise refocusing, so we can get a sharper phase image for those at the periphery. So, one of the power for phase imaging is really the non-invasive way. You can actually capture the cell dynamically for days or even weeks without interfering the regional function. So that's basically what one of the targets we're looking at drug resistance. So the first one, I give you an idea. The first one here, that's basically the cells that's growing. This is a 2.5 minutes per frame. We capture it over a 6 hours period. So you see the cells. There are some local movement. Occasionally you see some division, cell divisions, but overall the cell doesn't move dramatically. And this is the cell undergo a very high concentration of chemotherapy drug. So if you look at, pay attention to that cell on the top upper left corner there. So you see here the cell, the flat ones, and then suddenly they die. So you see all the blebs formation, and those are rare events. So if you capture, you can see some cell divisions as well. One thing that surprised me is the cell division, once that happen, they happen within minutes, like really fast. So if you use a scanning approach, you probably miss some those rare events. So the last two are those cells undergo very slow, so we give them very low concentration of the drug, and then they develop a resistance phenotype. So eventually the cell, this is actually 3-4 months of of the development. Eventually the cell will develop this drug resistance phenotype. So first you just look at the morphology, how dramatically different they are compared to the controls, and you see those extrusions of the cell. And if you look at them dynamically, actually you see how that dynamic, that extrusion of the cells in the cytoplasm, they move around and then they kind of, to sort of interact with the neighboring cells. And then also at the edge of the cell, the cytoskeleton, you see how they're actually quite flexible, a very dynamic process. And again, if you look at again, just look at the morphology of the cells, how much variation you see these cells is constantly changing in that process. This is all on the same time scale. So that's kind of give us the insight, how this dynamics changing over time. And these are with the phase, because you can capture those information quantitatively, so you can see all these vibrations in the normal cells, and these are the ones that's treated. You actually can see it's damped down a little bit for those that undergo kind of a cell death. And here is the one that you see the drug resistance ones, actually the opposite. You see how much fluctuation they have of those cell morphology. So all of these is, these are again another physics kind of characterization, but what does that mean biologically? So so what we do is we fix them, the image at that time point, and then do a hyperplex fluorescence to define the cell state. So we can correlate the morphological changes and all the dynamics prior to the fixation, and then correlate with sort of the cell state that defined by this sort of the multi-channel fluorescence imaging. So this is now more routine. So that is for the cross model, fluorescence versus phase. And here can also do the cross scale. So this is the phase image correlated with the super resolution. And the phase give you the nanoscale information in terms of the overall morphology, but the super resolution give you this sort of the molecular scale distribution of the molecule. So it's a chromatin with the nuclear periphery. And then if I zoom in this, you can get sort of appreciation of that cross-scale information between the nuclear membrane and the chromatin. They separate by roughly 40 nanometers here. So basically, this sort of cross-scale, what we want is we can capture this large-scale fluorescent image, and then identify through image registration, we can get this multi-model information combining the phase together with fluorescence and the super resolution. So these basically, you put that molecular scale information back into the context of the entire image. So it's fully automated, so it's a cross scale. So this, again, this is another example of looking at tissue, and if I zoom in that specific area, so this is the H&E correlated with quantitative phase and fluorescence. So the idea here is we want to take advantage of the standard pathological information with the phase that really give you the overall architecture, and then couple with some of the molecular scale information to learn about, this is a patient with inflammation. So again, this is the immune cells, so you see various type of contrast, and especially with the phase image, you see the extracellular matrix and some of the highlights, the higher value that come from the immune cells. And this is now more routine that people can do. So we developed that capability to do multi-cycle imaging. And this is a 4-cycle. We can get a sort of a complete, not really, never a complete, but more comprehensive information about the molecular environment of the tissue. So to analyze this, these are different types of immune cells. And then the H3K27 trimethyl is a marker for sort of higher value means it's more inactive. So you can see that inflamed cells, that indeed the value is lower. But with phase, you can also see those are label-free. You can also see that difference that indicate inflamed cells that have more open chromatin. So as a result, the refractive index is lower. The average refractive index is lower. So just conclude my talk, sorry to run over time. With the cross-scale information that you can really, the key is you can connect the information, putting the molecular scale information in the context of the systems level. And they require a multidisciplinary approach from instrumentation to the artificial intelligence for large-scale image analysis to robotics and automation, so you can really reduce the labor-intensive nature for the staining process and the imaging probe that can target low abundant target. These are, all the people that actually did the work, all my collaborators and funding support. One last thing, I'm actually moving to University of Illinois, Urbana-Champaign next January. So I'm looking for grad students and postdocs. So in various areas of instrumentation development, image processing, informatics. So any of you interested, you're welcome to contact me. All right, thank you for your attention.
Fantastic talk. Elana had to go to class, so don't take it personal. We don't have time, but if you have some questions. But that's for you.